Hello everyone, today we'll discuss the part 51 of the important notes for biochemistry so that you can easily able to answer the one-liners and the MCQs in your exam. Now first of all, the karyotyping. Now what is karyotyping? Now karyotyping is the process by which photographs of chromosomes, as you can see over here, photographs of the chromosomes are taken in order to determine the chromosome complement of an individual, including the number of chromosomes, and to find out any of the abnormalities in the chromosomes. Okay. So karyotyping is the best technique for detection of the monosomy or the trisomy, but can be done only during the metaphase of the chromosomes. Karyotyping cannot be done in any phase of the cell cycle. It cannot detect the amplifications, microdeletions, and complex translocations, but this karyotyping can only determine the chromosome complement of an individual, including the number of the chromosomes and any abnormalities over here in the chromosomes. Now the fluorescent in situ hybridization. Now this is actually a rapid or fast technique which can be done in any phase of the cell cycle. Fluorescent in situ hybridization can be done in morphologically intact cell or tissue or organ. Now this fluorescent in situ hybridization can detect the monosomy or trisomy, even the microdeletions, amplifications, and the complex translocation also. Fluorescent in situ hybridization tells the gene location on a chromosome. Now, restriction fragment length polymorphism or the RFLP is actually a lengthy procedure and can detect only the single mutation and those mutations which are actually affected by the palindrome. Probe is actually a DNA sequence only which is complementary to the gene of the interest or only a small portion on gene of interest. Probe is actually labeled with a radioactive material or a fluorescent dye. Hybridization or the blotting is actually used for the visualization of the sample DNA. Now the blotting techniques. Now first of all we have the southern blot. Southern blotting is actually a technique for detecting the specific DNA fragments in a complex mixture. Okay, and over here the sample analyzed is the DNA. The probe used is also a DNA, and over here the gel is also used. Now next is the northern blot. Northern blotting is used for detection of the RNA fragments instead of the DNA fragments over here. Okay, and the sample analyzed is the RNA instead of DNA. The probe used over here is the DNA and gel is also used. While third is the western blot. The western blotting technique is used to detect a particular protein in a mixture. Okay, so sample analyzed over here is the protein. Probe used over here is the antibody and gel is also used. Now southern western blot, or we can say southwestern blot we can say. Now over here the sample analyzed is protein DNA. Okay, the probe used is the DNA and over here remember gel is not used. Now next is the microarray. Sample analyzed in the microarray is messenger RNA or the complementary DNA. Probe used is the DNA over here and the gel over here is not used. Now next is the ELISA. ELISA, that means it stands for enzyme linked immunoassay. Okay, and over here the sample analyzed is the protein or the antibody. Probe over here used is the specific antibody for the protein to be detected and the gel is not used over here. Now single gene expression analysis can be done by both northern and the western blotting. Now in microarray or the small chip, okay, we discussed about this in our previous video also, that small CHIP chip, that means a microarray. While the capital C, small s, capital I, capital P chip, it is the chromatin immunoprecipitation. Okay, so over here in the microarray or the small C chip, small DNA fragments or the probes are immobilized on a solid support. Okay, for example, glass slide, okay, in an ordered fashion. Microarray can detect the multiple mutation. Over here, this can also be used in the multiple gene expression analysis or in the global pattern of the gene expression. Over here, this can also be used in the single nucleotide polymorphism or in the genetic transfer of the disease. Microarray cannot detect aneuploidy, that is monosomy or the trisomy. So today up till here, We will discuss about the further important notes in our next video. Thank you so much.